So if we think about what plants we need for the future, we need to think about what things are going to impact on agriculture. And I suppose future agriculture has to be agriculture that's adapted to a changed climate. And there's two broad ways we're seeing that happening. One is by developing agriculture that's more resilient to climate, but the other is to think about the location, where we're doing it. And we are seeing a future in which many crops move to quite different locations to where they're grown today. Another and contrasting type of response to climate change is that we're seeing at least rapid plans for moving a lot of agriculture into protected environments. And this is particularly true for high value products, and there's a lot of advantages in going down this path. And what will be challenging for us as plant scientists is choosing or picking which of the crops are going to stay out there and fend for themselves in the environment, and the ones we have to adapt to that changing climate, and which ones we're going to decide that's all too hard and we're going to bring them indoors and grow them in a controlled environment. So there are these two parallel futures for agriculture, and that's one of the big challenges for plant scientists, really understanding for particularly some crops which of those options will be taken. It applies much more to the high value horticultural crops than the field crops, but there are some field crops that will now qualify for this sort of treatment. The other overarching thing is, of course, that future agriculture will need to address consumer demands. And I always think that it's important to understand that there is about 5 different dimensions to the demand of a consumer for food. And roughly in the order I've got them here, the taste is pretty important. People are not going to eat something that they don't like the taste of. The convenience comes pretty close after that. It's got to be actually easy to eat, easy to prepare, and so forth. Health then becomes a consideration. I want to eat something that's not going to harm my health. In fact, I might like to eat something that will be positive for my health. Sustainability is becoming more of an issue. People want to think that their food is being sustainably produced and is not having an adverse impact on the environment. And then beyond that, they're actually concerned about ethics. That includes that slave labour wasn't used to produce the food, for example. So these are 5 very distinct drivers for consumers, and I think all of them have to be considered in defining the foods of the future. But the other one that's the really vague one is that we know that future agriculture is going to be impacted by new technologies that we don't even know about today. And that's we know about the great advances in information technology and biotechnology and nanotechnology, but it really is hard to predict how they'll play out. But there is technology push that does and will impact upon agriculture, and there's many examples in our life today of things that didn't come because somebody sat down and decided we needed it. It came rather because someone had developed the technology to do it and found an application. So we have to accept that agriculture is going to be severely impacted, I think, by technologies. And if I go through some of these, first of all, thinking about climate and agriculture and adaptation. I'm involved in organising a regular meeting now in Europe, and we're having the 3rd Agriculture and Climate Change Conference in Hungary in late March. And this conference is really focused very much on adapting agriculture to climate change, but increasingly we're seeing people start to think about whether we should also be talking about mitigation, because agriculture not only just needs to respond to agriculture, but it actually has an active role to play, because we can do things in agriculture that contribute to mitigating climate change, and that's a quite distinct option that I think we need to be aware of. In terms of trying to understand the genetics of plants and how we might produce plants that are adapted to future climates, I've worked with a number of model systems in the wild. This one I think was particularly useful, where we looked at wild barleys growing across a steep environmental gradient and asked the question, how are the plants adapting to those distinct microenvironments? And that's told us about the sorts of genes that plants use to adapt to a hotter or drier environment when we're working with a free mating population. We've also been able to do that with transects of plants growing sort of from Melbourne up across the Alps and down to sea level again. Again, we've been able to identify those genes that vary in those populations that provide the adaptation to those different environmental conditions. And so from this sorts of studies we can get clues of how we might have to change crops for new climates in the future. One of the things that's very much a work in progress is that we're looking at the dynamics of the rainforests in Eastern Australia. We're now beginning to look at the genomes of large numbers of species along the east coast rainforests up into Asia. So what the questions we're asking there is how those species are adapting to those different environments and how are they changing. What we've seen since Australia and Asia collided about 5 million years ago, because they were well separated prior to that, is that the flora have mixed. So we're seeing many species move down into Australia and others move up. And all of them have a different history. One of the interesting observations that you may have heard of is that Maurizio Rossetto, who's our key collaborator on this, has identified that some of the species have had human involvement in their dispersal within Australia, and some of those thousands of years ago. The classic example is the black bean. This is a very large seed, and the only vector that we can identify that could transport that seed is a human. No animal can carry it, and we see that humans have transported that thousands of kilometres and re-established populations probably mainly about 3,000 or 4,000 years ago here in Eastern Australia. So my big picture look at this is when I wrote this book in 2010, trying to identify how we utilise plants and rationalise the use of plants in agriculture and food production with their industrial use, and how we also reconcile that with conservation of biodiversity and the needs of agriculture to be developed in such a way that it's compatible with biodiversity conservation for wider environmental objectives. But much of what my group's doing now is really attempting to capture that biodiversity for use in agriculture. And we've systematically looked, we've done reviews now of exactly what uses people have made of plants across the whole tree of plants. Within the flowering plant families, there's a remarkable diversity of families that people have found uses for. And what we're now doing, of course, is getting genomes of all of these species. So we're beginning to understand the genetic variation across all of flowering plants as well. And a lot of that's driven, and I think it's important to understand, it's still driven by the needs for ongoing innovations in information technology. The China National Gene Bank is a really great example of the sort of iconic project that's attempting to capture or digitise all life forms. And we're collaborating quite strongly now with this organisation to try and look at how we can extend what they're doing in China to a global initiative on capturing all of biodiversity. If we're looking to the future, I like to always think what we can learn from the past, and so I've been interested for some time in how agriculture began. How did we first use plants, and how do we make that step, and what does that tell us about how we can reinvent plants in the future using those examples? And this is wild barley, Hortium spontaneum, photographed it here in Aleppo. It's growing there on the walls of the citadel. I think that was before it was bombed recently, so it doesn't look quite that good today. But this plant, we believe, was the beginnings of agriculture. I like to think this is where the internet started, because if we hadn't developed agriculture and human settlements, we would have never had the sort of social development that led to the internet. So it all began with barley. And this happened 10 to 12,000 years ago. We're fairly comfortable about that. And this was the first plant, I think, from many lines of evidence that was domesticated. And there's been lots of controversy about its origins. And just recently, in collaboration with our colleagues in China and in Germany, we've undertaken this large study where we've sequenced barleys from across their distribution, and particularly looked at barleys at the centres of origin in the Middle East here and in Tibet. There's long been controversy about whether there was a separate domestication of barley in Tibet, and in China it's been more difficult to access those samples because of the remoteness and the challenge of getting to material. But for the first time now we've had the data, and this is been an exercise of very large-scale data analysis. And what we've, to cut to the result, what we found was that barley was in fact domesticated originally in the Fertile Crescent back 10 to 12,000 years ago. The wild barley was then, the domesticated barley then spread both east and west, and we've been able to determine the route because we've been able to sequence with the Germans barleys that are 3,000 years old, so we can actually see 3,000 years ago what the domesticated genome looked like, with very high quality genomes going back to those ancient samples. And then there was a genetic narrowing as barley was taken up into the high altitudes of Tibet, adapting it to the cold. Relatively few genotypes were able to cope with that cold. So the modern Tibetan cultivated barley is a descendant of that process, probably about 4,500 years ago, coming across through India and up into Tibet. But then we have the wild barleys in Tibet that we originally had thought had been the source of the domesticated barleys there. But what we can now see by having the genome sequences is they're feral domesticated barley. So the barley escaped from domestication into the wild through back mutations that introduced shattering again. So while our early study, and I had a number of PhD students worked on it, that showed that the Tibetan barley was very closely related to the wild barley in Tibet, suggesting very strongly a local domestication. We can now show, by having the more detailed information that the whole genome sequence provides, that it happened the other way, that the order was such that the wild populations have arisen from domestication. And that's actually something that we now see also probably plays out for things like rice. Many studies published in Nature report on where barley was domesticated in China by comparing the wild and domesticated barleys at those sites. Probably all that they've established is that's where barley most recently escaped from domestication into the wild, rather than the flow being the other way. And so it's easy to be fooled if you don't really look in detail at the sequences. So we're really looking at reinventing crops like rice by going back to the wild and looking at how we can capture new diversity that allows us to adapt to new climates, that allows us to adapt to new demands by consumers, that it produces the sort of varieties that we need for the future. And there's a lot of diversity to work with. We're working with across the whole genus, and we recently reviewed all that is known, and rice relatives are pan-tropical. We're particularly focused on the ones in our region, and our region is from North Queensland to China and India, but they really extend all around the world. And what we've found is that the ancestors of domesticated rice occur in this area from probably just south of Townsville up into northern China and across to India. And that the Australian populations are the most diverse and most interesting. It's still unresolved, but if you look at the probabilities, that's more likely that rice moved from Australia to Asia than it moved from Asia to Australia, if you look at where all of the diversity actually is. So we've now reached the stage where last year we were involved in a major publication that really provided now reference genomes for nearly all of the species in the genus. So the objective with ERISA is to make it equivalent to Drosophila in the animal world, where it's a model genetic system where we have a sort of complete set of information about the whole rice genus. And we really are now thinking about that pan-ERISA genome that allows us to have pretty much complete knowledge about every variation in every rice that we've got to work with if we work with the natural systems. Big slide in terms of data. So what we've done very recently, this is unpublished work where we've looked at the whole chloroplast genome of 3,000 rice varieties. So it's a fairly large data set. So we've done a complete assembly of the chloroplast genomes of all 3,000 genotypes, and we've then done a phylogeny. So this is a very big phylogeny by any phylogenetic analysis that takes an extraordinary amount of computing time. But what we've got for the first time is an understanding of the diversity in the chloroplasts across all of rice. And what we see are two main groups. The colours represent the modern genotype, and what this shows us is that rice is very mixed up, that modern breeding has moved genes around, and we've got the so-called indica and japonica types, and the indicas are all black here, and the other types are japonicas. These yellow ones are basmatis. Here we've got tropical japonica, temperate japonica, but we can see that many of those actually have indica chloroplasts. So modern breeders have actually moved chloroplasts between all of these types. The type is probably based on the nuclear genome type, but the chloroplasts have moved around and been quite scrambled, and that's something that we didn't know before. But there are actually two basic types of chloroplasts in domesticated rice, and they probably represent those early domestications of indica and japonica. So they still trace back to those two wild types that we can find. We're also coming to understand that there's multiple origins of things like the Aus group. So what this analysis does is now begin to give us much better understanding of the relationships across all of rice. And here in Queensland, we're working with collections now, extensive collections we've got from all of the north, particularly in the areas of greatest diversity in the Central Cape, but right up there. Rice doesn't occur right to the tip, but it goes almost to the tip of Queensland. And this is allowing us to understand diversity of rice in the wild. And what we've really understood, what we've discovered for the first time that's really exciting, is we've discovered natural hybrids between some of the rice species. And this, I think, is really central to some of the events that led to domestication, and that will help us to understand how we can restructure rices for the future. So in Australia, we've got 4 major species. They're all interfertile with domesticated rice. They represent the primary gene pool. They have great resistance to Australian pests and diseases, so they provide a key resource for adapting rice for production in northern Australia. And they have some really novel nutritional traits which we believe will be really strong selling points, and Bob here is helping us to unravel some of those. So there's a real option here for having a highly productive rice industry based on this native plant that provides the right sort of environmental resistances, but provides new characteristics desirable for consumers. One of those is grain size. These bigger ones here are some of the local Australian ones. This is what you see in an Asian rice. So we've got genes for grain size. These are single genes we've now characterised. So we've got, for example, a gene from one of the northern species that we've back-crossed into domesticated rice, and gives us a 20 or 25% bigger seed. So that's not only an attribute that potentially contributes to yield, but it also contributes to consumer attributes, because in a number of markets people like big rice. They like the appearance of it. So that's a trait we hope to work with. And also, of course, we're interested in studying the starches of these, really quite novel starch characteristics which should be desirable. One of the features of rice that I've been involved with is fragrance. Fragrance approximately doubles the value of rice, and it's due to a gene that we discovered back in 2005. One of the first really significant discoveries to come from the rice genome sequence, and we actually got to this point before the public release of the genome sequence. And this mutation leads to the fragrance of the rice, but also leads to a loss of function of a stress response pathway that's really critical. And all of the fragrant rices in the world, even though they're high value, are very hard to grow at high yield because they're susceptible to stress. And we've been trying to work out how to compensate that for some time, but we've had a significant sort of breakthrough in working in Pandanus. Many of you will know that Pandanus, the pandan, is the source of the same fragrance. It's used to flavour rice. People put the leaves in the rice to get the same flavour. We had a series of scientists working in the lab and have identified that it's the same mutation in the same gene. But what's curious here is that Pandanus grows at the beach where it's quite salty. It's very salt tolerant, and the fragrant ones are extremely salt tolerant, whereas fragrant rices are incredibly salt susceptible. Well, what we've found by looking at the transcriptome of Pandanus is that there's a different pathway that bypasses the blockage that's caused by this mutation. that allows us to respond to stress. And so these plants can be both salt tolerant, stress tolerant in terms of abiotic stress, and produce the fragrance. So this gives us a new option for engineering high-performing fragrant rices by taking the chemistry we've discovered in Pandanus back into rice. And this is a pathway that involves some novel amino acid metabolism. So we're looking at doing many of the things we're doing in rice, and at rice we're getting much closer to having products. In sorghum, we're currently starting to sequence many of the genomes of the more exotic wild sorghums further away from the gene pool. But of course, sorghum is essentially an Australian genus. You might even say it's a Queensland genus. The one that we domesticated was from Africa, but all of the diversity, all the high-level diversity is found here. And wheat, we don't have anything close to wheat, but we've actually got quite a lot of distant wheat relatives that probably are sources of quite interesting genes. So we're going to look at some of those. As sources of genes, we can't necessarily cross these in the same way we can with the sorghums and the rices, but they'll be important sources of genes. In wheat more broadly, by particularly looking at the transcriptome, we've made what I think are really major discoveries that support wheat at three different levels. And this has come really from studying the domesticated gene pool. Whereas in things like rice, I think all of the interesting diversity is still to be captured from the wild. In wheat, the domesticated gene pool really has a remarkable amount of diversity, much of which hasn't been exploited and isn't found in our commercial cultivars, but it's there in some of the other material that we've got available to us to breed from. We've discovered, first of all, a gene that determines the low volume in bread. So it's really a single gene that really explains almost all the variation. We don't need complex selection here. There's just one allele that seems to be associated with good low volume. We've discovered that flower yield is largely controlled by a couple of genes that are cell adhesion proteins that hold the grain together. By downregulating those, we get more flour. And this explains pretty much all of the variation we find in wheat. It's much simpler than we thought in the old days of QTL analysis and so forth. When we look at the actual genes, we've got a simple story. And in the grain, we have found novel pathways that suggest carbon metabolism pathways that really provide some of the explanation of why wheat's a successful crop in temperate, dry environments, that it's able to continue to capture carbon under more stressful drying conditions than would be otherwise found, and then is possible in grains like rice. So looking at the transcriptome of the wheat grain, we've been able to identify genes that influence the amount of grain we get, the amount of flour we get from the grain, and the amount of bread we get from the flour. Putting these all together, we get a fairly substantial increase in the amount of food we can produce from wheat. And put together, these really represent a significant opportunity to remove the barriers to genetic improvement in wheat globally, because some of these quality constraints have really limited the rate at which genetic gain can be made in global breeding programs, and really they can be largely removed by selection at just a couple of genes. We're also starting to work more widely with the pulses, and I think this is an area where there's real opportunities to invent completely new pulses for the future. We're sort of stuck in a rut. We've got the various pulses we've got now, but if you look across Queensland, there's a lot of species there that really look like they wouldn't be bad crops and really could represent something quite fresh and new, as well as having, of course, here in Queensland, all of the diversity in the Glycine, the soybean genus. Australia is really where the diversity is for soybean, even though it's, in the case of soybean, much more distant from the domesticated soybean than it is for things like rice and sorghum. But also the vignas. There's tremendous diversity of vignas here. And if you go to the herbarium, it's full of Glycines, the soybean relatives, that are unnamed. There's just so many new species there that we don't even know how many species there are in Queensland. It's really an area that's got a lot of potential. With Pomelo, we're working on the Cajanus, back-crossing the insect resistance into pigeon pea, and that looks like it's going to work. So we've got wild material. The centre of diversity for this genus is probably Queensland. This is pigeon pea. We've got wild material that's really going to be essential in adapting this species to local production, and we're moving quickly to utilise it. In the horticultural crops, we've got a new program, a horticultural tree genomics program that's focused on these 5 species, avocado, mango, macadamia, almond and citrus. And the reason we're working on those 5 is because these represent the bulk of the value of horticultural tree crop production in Australia. And so this, by doing detailed genomics now on these species, we're going to be able to lift the productivity, hopefully, of these species over the next 5 years. And this will support, for example, the small trees program that's been going on to try and, again, reinvent these crops as more intensive production systems. And we've had many successes at working at genomics in fruit. Comparative genomics of these 3 species allowed us to, just by comparing the genomes at the species level, to identify many of the key genes important for many of the traits that distinguish these species in production. The macadamia is one of particular local interest with these 4 species, and here we're hoping to be able to really provide a new platform for genetic improvement. This species here, Macadamia jansenii, is quite rare, although there's just been reported a new population of another 37 plants have been found in the wild. But here we've got a chance of not only having the genome of the species, but having the genome of every known individual. Because these rare species, we can sequence everything. And so these species do represent trees that will offer important traits to lifting macadamia production. And that's something that's particularly local, given that the whole diversity is north and south of where we are here in Brisbane at the moment. In the Proteaceae, which is the family, the Southern Hemisphere family of which macadamia was is found, there was a national competition to nominate species for genome sequencing for Bioplatforms Australia. And I did nominate the macadamia, but I was unsuccessful. The waratah won, and so we're now sequencing the waratah, and one of the rationales is that it will provide a model system for macadamia in the Proteaceae, that we've got a plant here with a much smaller generation time that will certainly be able to use some of the information from the waratah in the macadamia work. But the waratah, I'm now going to be coordinating a project where we look at diversity in the waratah. It's been collected over a very wide range of sites, and it represents a species that has both inbreeding and outbreeding populations. So we can begin to understand what drives those different reproductive behaviours at the evolutionary level in the wild populations. And it's a plant of some economic importance, and it's the one that is now nationally funded for the first genome. The next steps in this program will be to think about identifying a species in every genus of Australian plants to sequence those. So this is really a pilot for what's seen as the next stage, where we go to genus level sequences across the Australian flora. Citrus, this is a Citrus australasica from my garden. This species is now being used as a parent in breeding citrus here in Queensland. There are some really interesting traits in the Australian wild citrus, and things like flowering time. So early fruit, which is a really important characteristic, is a trait that will be sourced primarily from Australian germplasm. So even though these things don't look particularly suitable, they have a lot of genes that are not only potentially useful, but are actually now being used. Coffee. Inventing coffee for the future may be a challenge. I was at the PAG meeting in San Diego, and I had a call from a journalist in the ABC who interviewed me about a report from Kew that coffee was going to become extinct in the wild. There's There's about 100 species, but most of them are actually endangered. They all grow with rainforest understory in places like Madagascar that's largely all been cleared. There's no habitat left for many of these species. And we've been looking, and we've actually done a recent phylogeny of the whole genus, looking at all the species and their relationships and identifying some new principles about how we do those types of studies. And there's some fairly diverse things. This is one from North Queensland, up on the Cape, an Australian species. And we've really identified that this is a really interesting case because many people seem to be not that concerned about climate change, but the response to the story about coffee was that there was enormous interest in the thought that climate change might mean that coffee would be not available to people. And so the ABC has come back to us several times asking whether we get any more stories about coffee because it's the most popular story they ever have run on their science website. So we now have some new clues about how to communicate the importance of climate change. We have to think about something that people care about. Maybe coffee's the way to get some action on climate change. Bing Chen has done a great study where we've actually looked at using the transcriptome what happens at different places in the canopy, and we now understand what controls coffee quality, and we can explain really why those high altitude environments in the tropics produce those high quality Arabicas. It's all about slow development of the berry. We need to get high sugar content, high caffeine content, and that happens lower in the canopy where it's slower. It happens less so on the upper canopy, so we make better coffee from the beans at the bottom than we do at the top. We can explain that now in terms of gene expression, and we can think about what that means for producing high quality coffee into the future. I think we can define environments in which we can continue to do that, produce maybe here in Queensland, in controlled environments, high quality coffee by knowing what the environmental triggers are. And we've actually now identified the control points in the caffeine biosynthesis pathways. We've explored several hundred coffee genotypes across all of the diversity, and we now are able to, I think, genetically manipulate caffeine content, sugar content, all the things we'd need to do to invent a coffee that any particular consumer might want and understand how to produce it in a particular environment. The one species that I'm particularly keen on reinventing is sugarcane. I think that we really need to think about this as a sugarcane crop. The local industry for a long time has thought of itself as a sugar industry. I've for decades been trying to convince them that they're a sugar cane industry and that the products that can be produced from this plant are quite diverse, and sugar is only one of them. The fact that we now face quite a crisis, I think, in the sugar industry. The price of sugar is low, and I don't think it's ever going to go up very easily because world demand for sugar is finally declining. I think people all around the world are turning away from sugar for health reasons, and so the relentless rise in sugar consumption has finally stopped, and we're seeing a decline in sugar consumption, just at the time when the world's developed even greater capacity to produce even more sugarcane. So the production of huge quantities and the drop in demand for sugar certainly means that the sugar price is not going to be attractive. And so we've had a long-term effort of trying to think about how we can use this incredibly efficient plant to produce other products, that more valuable products and things that might have a longer-term future than sugar. It's an incredibly complicated genome, and I now think we want to go from sugarcane to something we might call new cane. This is a cartoon of the genome, very complex, more than 100 chromosomes. We've got to the point where we've got a monoploid reference genome. We're midway in generating a fully polyploid genome with the Joint Genome Institute in the US and our collaborators in France and CSIRO. And we would think that probably by the end of the year we'll have working drafts of the whole sugarcane genome. So this is the last of the top 20 crops in the world to have its genome sequenced. And the reason it's the last is it's the most complicated. And it's still very challenging. Adam Healy will give the seminar here next week. Adam was a PhD student in my group. He now works for Hudson Alpha, and he worked here on the eucalypt genomes. But in the US, he's been assigned the sugarcane genome assembly by JGI, and so hopefully he'll give us a bit of an update next week. One of the things we've been doing is sequencing individual chromosomes. Because we have so many chromosomes that are very similar, We've resorted to actually isolating the individual chromosomes and sequencing those. It's technically demanding, but it's one way that we can overcome some of the bioinformatics challenges of assembling such a complex genome. And in terms of diversification, the driver for some time has been energy security. Australia's become much more dependent on imported oil, and I've had a lot of engagement with the US, particularly the Navy through the Pentagon, about how we can produce more fuel here to support the US's operations in the Western Pacific. But really, I see that the business is moving on. That we've been working on how to convert sugarcane bagasse, this is a sugar-free sugarcane if you like, or one that the sugar's been removed from, to sugars by series of biochemical processes, with the view that that can be converted to a range of fuels. We can do this very efficiently now from sugarcane bagasse to ethanol, and that's what we do routinely at the lab level to test the various processes and the various types of biomass that we generate. But really, I'm now an electric car driver, and I just don't see a future for liquid fuels, not beyond 10 years or so. So I think that the real role for this is in biomaterials and plastics, fabrics, building materials. So if we look at the use of oil in the world, about half of it goes into transport fuel, and the other half, or more than the other half, goes into these other products. And even if we don't need biofuels or plant-based carbon for fuels, if we're going to replace that with electricity or other sources, we still need the carbon for all the other products. We need the carbon to make these carpets, to build the wall panels, and all the other things that we make from oil today. And so we're positioning sugarcane as the base of industries that would produce all of those products. And they can flow through the same processes that we've been developing, because they really can be made from sugars, or they can be made directly in the plant. And there's quite a wide range of feedstocks, industrial chemical feedstocks, that we can now make directly in sugarcane. And so in achieving this product diversification, we've been working directly with the sugar mills. This is the Mareeba mill, back before it even made sugar. At this stage, it was just crushing juice and shipping it to the coast. We're working now with a number of mills on trying to work out how they can reinvent themselves to make things other than sugar, and how we can provide them with the right plant feedstocks that optimise that transition into a future where they're not dependent on the price of sugar. And I think this is going to be, for Queensland, a really important step, because sugarcane has been such an important crop here in Queensland. So I don't think sugar is finished, but I think sugar is just a byproduct now. We'd really have to think of other things being the main products we make from this very efficient crop. So this biochemical route, we've really worked fairly hard through. We've had the Smart State project. This is where it allowed us to get the Joint Bioenergy Institute in San Francisco as a partner, and Sugar Research Australia, of course. And we really do, I think, now understand a lot more about how plant biomass variation can facilitate the more efficient conversion through these processes. A lot of it's about doing biomass chemistry, and some of that's now expanding. We recently had a UQ deal with IIT Delhi, and we've got a student coming to work on how we convert, how we produce aromatic compounds from the lignin components within the sugarcane biomass. So we can look at an expanding array of high-value products produced from this crop. We've also done quite a bit of work on eucalypts in this context, because eucalypts are one of those other sources of biomass that we could produce and might complement sugarcane production, particularly in North Queensland, to extend and allow us to have biomass available for chemical production all year round, rather than just when there's sugarcane available. And we see lots of interesting things. One local plant that produced some interesting opportunities for us in terms of reinventing plants is the corymbias, the spotted glums locally. This is variegata and henryi, two very distinct species that grow locally here within the Brisbane city limits. One's a very big tree with big leaves, the other's got small leaves. All of our analysis suggests that this is probably a single species or a single population segregating for maybe one allele that determines the size of the plant. So we're very keen to identify that, because here we've got a species where the foresters tell me they have problems with contamination. They plant the seed from one and the other one, some of the others come up in it. What it really has told us is that this is a single species varying for a trait that has a really remarkable impact on its morphology, such that the taxonomists are convinced they're completely distinct species, whereas at the DNA level there's no difference. Or probably, maybe just a single SNP. I wanted to mention Jerboisia. We've got a strong and growing program on Jerboisia. Jerboisia has been used in Queensland for quite a long time to produce high-value alkaloids as pharmaceutical. We're reinventing this crop again by looking at all the species, going back to the genome, looking at understanding the pathway of production of these high-value compounds, and looking to produce plants with higher levels of the product. This is already $1 billion industry here in Queensland, but we think it can grow substantially. So these products are used for travel sickness control. They're used for post-operatively. If anyone's ever had a GA, they've probably had some of this to control nausea. Very widely used globally. Natural product, sourced all from Queensland because this is where these plants, only place where these plants grow. But we've got an opportunity here to reinvent them using, I guess, modern science, because the current industry is based on material that was domesticated in the 1920s and 30s. Jojoba, this is a plant from the Arizona desert and into Mexico. We've got some students here from Saudi Arabia working on this, and we've got the Vice Chancellor from the university coming tomorrow to sign an agreement with our Vice Chancellor about studying the jojoba genome and identifying the genes that control oil content, stress tolerance, and of course the sex. These have got male and female plants, and we think this will be an ideal system in which to determine the genetics of sex determination in plants. So that's an interesting new venture, and we expect to have maybe a group of 6 or so people working on this system. So I proposed a research hub, which we certainly haven't heard about, but which would allow us to work on all of these species from rice to citrus and really explore more extensively how we can mine this great diversity here in Australia and make it available for agriculture and genetic improvement globally. I think we've thought that Australia was a poor source of genetics for agriculture because we were really looking just in a very narrow sense at the plants that had been domesticated thousands of years ago elsewhere. But when we look at what we can do with current technology, getting out into the secondary and tertiary gene pools, Queensland and Australia are very rich sources of genetic diversity for very many of the really major crops, and really are the key places for rice, sorghum and soybean, and also pigeon pea. Globally, there's nowhere probably more important. So in addition, of course, to capturing those genes for current domesticated crops, we can think about completely new ones. And some of the ways that we're doing that is thinking about how we can take alleles or genes we find in the wild and introduce them directly. So we can edit a gene we find in the wild into a domesticated crop and immediately have that trait. That's something that wasn't an option years ago, and so now the way we're looking to screen this material is to screen for novel alleles and test their function by putting them individually into a domesticated background. That way we can very quickly evaluate what utility that gene might have if we were to introduce it into a crop in a commercial sense. And we've got some really novel variations here that influence starch properties in rice that we're actually testing by gene editing at the moment. So, Australia's got 10% of the grasses in the world, all of the subgroups, including many that look like they could be cereals if we wanted to domesticate them, adapted to a very wide range of soils and places. This is one that we've taken through the process to some extent, Microlitus dipoides, and that's a temperate adapted grass, so this is a potential to grow a rice-like product in cool climates in southern Australia. There's all those sorts of options available if you start thinking about these sort of strategies to reinvent plants for the future. We're working with the CRC in Northern Australia on rice, and we spent Thursday and Friday with them workshopping how we can develop a rice industry for Northern Australia. We've got all of the stakeholders now engaged in doing that, But one of the key elements will be to capture some of that novel diversity we've got in Australia to produce a product that really is differentiated, that is attractive to consumers because it's novel and Australian and has maybe desirable health attributes, desirable taste attributes, things that will make people choose this in the supermarket over other rices. And so we see a very positive future for rice as a new crop in the north. And we're hoping that the CRC, through this model, will see that there may be other options beyond rice that they can think about. I'm going to this meeting in Montpellier in June, the third Harlan Symposium. This is a series of conferences on this broad question on the origins of agriculture, domestication and evolution in relation to utilisation of genetic resources. The first meeting was held in the Fertile Crescent. The second one we held at University of California, Davis. The third one here is at Montpellier, and we've been successful in attracting the fourth one here to Brisbane to join TropAg in 2021. So that'll be an opportunity to get a critical mass of people interested in this worldwide to come to TropAg for 2021. And I'm going to Montpellier to announce that the place is Brisbane for the next conference. So I guess to sum up, if we think about where we're going with plant improvement technologies, because I've been involved through a lot of the iterations that have happened. If we think back, we started out with plants doing purely phenotypic selection. First, maybe 10,000 years ago or so, but by about 5,000 years ago, we were actually doing crosses. People were actually doing controlled pollinations, we believe, that far back. But really, modern plant breeding as we know it, started with Mendel and around that time in the late 1890s. So really it's a 20th century activity in the main. And out of that we got, of course, a whole lot of advances. The Green Revolution, associated with the semi-dwarfs, changing the harvest index. A lot of this was driven by application of statistical methods to control crosses and so forth. But then we had the development of molecular techniques, and it's hard to know when you want to say all of this has begun and when it's impacted. From the 1980s, we've seen marker-assisted selection begin. I think it really didn't start in earnest until after 1990. PCR was pretty important. So PCR's late 1980s. By the early 1990s, we're using much more widely markers for selection in plant breeding. From that, of course, through the same era, we saw transgenics. And we saw the advent of technologies like genomic selection. I think if we look forward into the future, certainly from the last few years into the future, I think we're looking to have a much more deliberate approach that we might call design breeding, or it's very much data driven, that we will actually work from having, the starting point will be having the sequence of all the individuals that you're working with. We'll be deliberately designing which gene you want at which locus by understanding the phenotypic implication and the breeding value of every allele, using extensively gene editing to get there, and particularly basing breeding on the knowledge of gene function and what a particular version of the gene will deliver for you. And that's really very powerful. I've seen recent analysis showing that by gene editing, we can really lift the rate of genetic gain. So it's because if you gene edit, you don't have to worry about linkage drag, you don't have to worry about imperfect linkage if you're using a marker. So increasingly people will look at a system, identify the 50 genes they want and edit all 50 of them at the same time. That's going to be technically possible. So you can actually decide what allele you want at each of those loci and just generate it. And you can do that fairly quickly. It doesn't involve years of backcrossing or something. It involves one generation of the plant. And I think that's the sort of future that we can drive by having knowledge of all this material, and that what we've got here in Australia is a unique biodiversity that gives us more options that we can test in this deliberate design breeding future. So I've finished with the usual ad for TropAg. Thank you.